cell-to-cell communication and development. Morphogenesis is the result of cell-to-cell communication through cell signaling pathways in the development of organisms. During morphogenesis, each type of cell in the developing embryo are arranged into distinct patterns to form structures of the tissues and organs because cells have different sets of proteins in its cell membrane. Differential cell affinity. Presumptive epidermal cells, dissociation of cells, neutral plate cells, spontaneous reaggregation will be taking place. Later segregation of cell types. Outside neural cells, epidermal cells will be arranged, then cross section will be taking. So we could see neural cells on the inside, then epidermal cells on the outside. The differential cell affinity in the embryo is primarily due to the presence of cell adhesion molecule cadherin. Cadherin structure, function and types. So cadherins are transmembrane proteins having a 5-domain extracellular end that interact with other cadherin homophilic interaction on adjacent cells forming cadherins junction and intracellular end anchored by a complex of proteins called catenins. So cadherins are calcium-dependent adhesion molecule. Therefore, the second domain of extracellular end have calcium binding site. The cadherin-catenin complex bind to the actin cytoskeleton of the cell, thereby providing the mechanical forces for forming tubes. In vertebrate embryos, several major cadherins types have been identified. E-cadherin is expressed on all epithelial tissues of embryo. P-cadherin is found predominantly on the placenta, where it helps the placenta stick to the uterus. N-cadherin become highly expressed on the cells of the developing central nervous system. R-cadherin is critical in retina formation. The class of cadherins called protocadherins lack the attachment to the actin skeleton through catenins. The timing of particular developmental events can also depend on cadherin expression. For example, N-cadherins appear in the mesenchymal cells of the neighboring chick leg before condensation to form nodules of cartilage. If the limbs are injected just prior to condensation with antibodies that block N-cadherin, the mesenchymal cells fail to condense and cartilage fails to form. Then cell migration. The first stage of migration is polarization, wherein a cell defines its front and back, directed by diffusing signals from ECM that recognize the cytoskeleton. The second stage of migration is protrusion of cells leading edge by polymerization of the actin microfilaments at the cell membrane, creating long parallel bundles. The third stage of migration involves the adhesion of cell to its extracellular substrates. Integrin spans the cell membrane, connecting the ECM outside the cell to the actin cytoskeleton on the inside of cell to form focal adhesion. Hemidesmosomes. Fourth stage release the adhesion by proteolytic destruction activated by Ca2+ ions and allow cell to migrate in forward direction. We were talking about N-cadherins appear in the mesenchymal cells of the developing chick leg before condensation to form nodules of cartilage. If the limbs are injected just prior to condensation with antibodies that block N-cadherin, the mesenchyme cells fail to condense and cartilage fails to form. Then cell migration. The first stage of migration is polarization, wherein a cell defines its front and back, directed by diffusing signals from ECM that recognize the cytoskeleton. The second stage of migration is protrusion of cell leading edge by polymerization of the active microfilaments at the cell membrane, creating long parallel bundles. The third stage of migration involves adhesion of cell to its extracellular substrates. Integrin spans the cell membrane, connecting the ECM outside the cell to the actin cytoskeleton on the inside of the cell to form focal adhesion. That is the hemidesmosome. Fourth stage release the adhesion by proteolytic destruction activated by Ca2+ ions and allow cells to migrate in forward direction. Then cell signaling, induction and competence. The precise arrangement of corneal and lens tissues in vertebrate size formed by coordination between cells, for one group of cells changing the behavior of an adjacent set of cells, thereby changing their mitotic rate. The kind of interaction at close range between two or more cellular tissues of different histories and priorities is called induction. The close component. The first component of inductive interaction is the inducer, the tissue that produces a signal, paracrine factor, that change the cellular behavior of other tissue. The second component, the responder, is the tissue being induced. The ability of a responder tissue to respond to a specific inductive signal is called competence. must have a receptor protein for inducing factor, the ability to respond to its signal. The two major parts of the eye construct each other, and the eyes form the reciprocal paracrine interactions. The head ectoderm is the only region capable of responding to the optic vesicle. If an optic vesicle from Xenophus leviscenterio is placed underneath head ectoderm in a different part of the head from where the frog optic vesicle normally occurs, the vesicle will induce that ectoderm to form lens tissue. Trunk ectoderm, however, will not respond to the optic vesicle. Only head ectoderm is competent to respond to the signals from the optic vesicle by producing a lens. One induction will give a tissue the competence to respond to another inducer. For example, the first inducers of lens may be the endoderm and mesoderm that underlie the lens forming ectoderm at mid-gastrula stage. So PAX6 is a transcription factor in the anterior ectoderm and act as competence factor and provide competence to respond to the inducers from the optic cup. Inducer molecules. The optic vesicle appears to secrete two paracrine factors, a BMP4, the protein received by the lens cells, and induce the production of XOS2-TF. FGF8 signal transducers, the appearance of the LMF-TF. So in the ectoderm, the combination of the TFs activate lens-specific genes such as delta crystalline. Types of interaction between cells on the basis of source and action of signaling molecules. Autocrine interaction. When the same cells that secrete paracrine factors, it has its own receptor to respond to the placental cytotrophoblast cell synthesized and secrete CDGF, whose receptors is on cytotrophoblast cell membrane, result in explosive proliferation of the tissue signaling molecule. Then paracrine interaction. Proteins synthesized by one cell and can diffuse over small distances to induce changes in neighboring cells. Example, vertebrate eye lens induction, ectoderm by optic vesicle cells. Then juxtacrine interactions. When cell membrane proteins on one cell surface interact with receptor proteins on adjacent cell surface, which is signaling pathway involved in neural development. Contact-dependent juxtacrine. Endocrine interaction. Cells producing signaling molecules and secrete into the immediate blood vessels and travel through this bloodstream up to the target organ and exert their effects.